Hello and welcome back. This lesson will discuss in more depth bacteria. Bacteria is a common and large problem when preparing food for the public or you. These precautions that you take when you're in a restaurant, you should be taking them using these precautions as well, right? I mean, it's good that you value their life, but don't you value your life and your family's life? So everything learned here should be applied at home as well. Bacteria do not wear white hats and black hats. You can say, well, it's bad bacteria. Is it really bad? Oh, you're a bad bacteria. Go to your room and don't come out till I tell you to. Are there any bacteria wanted posters? But bacteria sheriff? No. Bacteria is bacteria, and it exists for a reason, and it exists to grow and reproduce the same as you do. But it's not good or bad. However, when you're serving food, that if certain levels of certain bacteria are in your food, then it's a problem. And the better you understand bacteria, the better you will be able to prevent it from being in your food and how to address it. Bacteria, they reproduce. So everything alive does, tries to. Bacteria are produced by cell division. One becomes 2, 4, 8. Actually, it's logarithmic. And so on. This doubling is called binary fission. Under ideal conditions, bacteria multiply at an explosive rate, which is what we're going to delve into. The growth pattern, lag phase, bacteria adjusting to a new environment, getting it together. Growth phase, once it, gets the hang of where it's living, you're adjusted to your new house. The growth is explosive. As the resources decrease, then the number of those born and the number of those die become equal. And then as they overconsume the resources, then they begin a death phase, which is a biological fact that humans are ignoring or choose to ignore. In the human body, in meats, plants, soil, fish, and water, bacteria more common than any other organism. What is biomass? Biomass means a biological carbon-based substance which meets our definition of being alive. Two main kingdoms, plant and animal. Now, if we take the total amount of biomass on Earth and look at how much of what, here's what we're seeing. The total weight of all humans on the Earth, their biomass, compared to the whole picture is 0.01%. Our weight is only 0.01% of all the biomass on Earth, roughly equivalent to the biomass of termites. I know that may seem strange, but it's a fact. Okay? We and termites, they're 0.01% biomass. We're 0.01. By comparison, bacteria is 13%. Can you see the difference in 0.01 and 13? Huge difference. There's a whole lot more bacteria than there is of you. Plants are 82% of the biomass, but mostly it's algae in the water. Now, since bacteria requires nutrients, as does any living thing to function, water, air, food, it can run into problems. Disease-causing bacteria, meaning a bacteria which can cause that substance a problem if it happens to wind up there. Pathogens feed on the nutrients. Well, it's alive. You have to eat to live. And potentially hazardous foods, meaning foods that if this bacteria is in, it's an issue. And they can multiply very rapidly at favorable temperatures, while other bacteria discharge toxins. As they multiply, die, go through their life cycle just like you go through your life cycle. Your small intestine contains over 400 different types of bacteria. If they weren't there, you wouldn't be here. Are they good or bad? Well, apparently they're good. But some bacteria, when they're there, is not good for this particular organism, being a human. Bacteria classification. First is their shape. And when you look under a microscope, they can be round, which is cocci, rod-shaped bacilli, look like little spirals, spirilla. A lot of them look like fat commas, vibrios, corkscrew, spirochetes. Now, oxygens have different, bacteria have different worlds that they live in. Aerobic, some have to have oxygen or they're out of there. Anaerobic, they can't be around oxygen or they're out of there. Facultative aerobes or anaerobes, they're more creative cousins. They prefer one, but they can deal with the other until hopefully their world improves. Aerobic bacteria can produce STP, which is the energy source of all animals, more efficiently in an oxygen state. But when they're in a non-oxygen state, they can go into fermentation cycle, which is less inefficient, but they generate enough STP, triphosphate, to survive until hopefully they can get into a better environment. E. coli is facultative aerobic. You have a steak laying in front of you. If E. coli is present in that steak, it will wiggle its little way to the outside of the steak because there is oxygen, and in the middle of the steak there's less oxygen. The steak sitting there, if there's E. coli, it'll be on the outside because it is a facultative aerobic bacteria, which is why whole cuts of meat only need to be cooked to 145 degrees, because if that bacteria is present, it'll be on the outside, not the inside. But if you grind that steak up into, say, hamburger meat, that E. coli will now be distributed everywhere within that meat. So that's why when you grind it up, you have to cook it 10 degrees higher, 155, because you distributed the E. coli throughout the meat product by grinding it up. Another classification of bacteria is gram-positive or gram-negative. Gram is the person who came up with this, so as usual in chemistry and biology, it's named for the wonderful person that came up with it. Bacteria shapes. In the upper left-hand corner, you'll see coccus or cocci, more round. Next to that, top line, rod, bacillus. Spirochetes next to it. Below that on the right-hand side, vibrios, spirilla. Those are by far the most common. The others are special cases, but in what we're doing, not necessarily important. Gram staining is a laboratory technique that when someone says, okay, what kind of bacteria is this? What do we do about it? One of the first things we do, we put it on a slide and gram stain it. It's a pretty easy process to do. Staining things on slides is done, very common in the lab, because different types of stains bring things out about whatever's on that slide. Violet dye is applied, and then decolored, and then a red dye. The cell walls of certain bacteria retain the first dye and appear violet, violet, and while those who lose it, gram-negative, appear red. You're looking at a gram-positive slide. What if you stain this slide? Yeah, this would be a gram-positive slide. Here's what a gram-negative bacteria looks like after gram staining. And here's one. On the left side, you see a bacteria that's gram-positive. You'll notice that there's only one cell membrane. On the right, gram-negative, you'll see two, cell membrane and cell wall. And if we know they're gram-positive, gram-negative, then we can quickly, categorize the bacteria, because we know which bacteria has a cell membrane only and which bacteria has a cell membrane and cell wall. Bacteria growth factors, FATM, FATTOM. Takes 6 things for this to happen. Food, proper acidity, pH, it can't do it instantaneously like most things in the universe. It has to be at the proper temperature for that organism to do this. It has to be an oxygen requirement. Try going into a no oxygen environment and living. And every living organisms take oxygen, oxygen, moisture, water. That's why when scientists look for life on other planets, first thing they look for is their water. That doesn't necessarily mean there isn't life forms without water, but life as we know it requires water. If there wasn't water in the earth, there would be nothing living on it that we can associate with. Food. Bacteria can grow at dangerous levels. High protein food. High protein, well, we grow best when eating high protein too, right? Meat, poultry, seafood, dairy product, cooked pasta, cooked beans, other food, plant origin that has been heat treated. Heat treating vegetable products or slicing them, you just defeated their immune system. Their defense system, which makes them vulnerable to bacteria you don't want there. And of course, then they will support rapid and progressive growth of bacteria that you're trying to prevent from the product that you're serving. Acidity, pH. I think we got into that before. Water 7.0 is basically seawater. The amount of hydrogen ions there. Bacteria grow best in food that is neither strongly acidic. When you have a jar of I don't want to say jalapeno peppers or something else that's highly acidic, a lot of vinegar in it. Does it need to be in the refrigerator? No, because bacteria can't grow in it. It's too acidic. Um, these potentially hazardous foods are more likely to spoil. 4.6 and 7. That means food prefers slightly acidic. Vinegar, citrus fruits make it difficult, but not impossible for bacteria to grow. Here's a chart of different bacteria and the pH where they can survive and grow. First one, Clostridium perfringens. You know what Clostridium perfringens is? Every animal plant that lives is full of Clostridium perfringens. It's a natural thing. When that organism dies, whether it's plant or animal, Clostridium perfringens multiply rapidly. When the plant or animal is alive, that plant or animal's immunity system keeps them in check. Can't get rid of them. It keeps them in check. When a plant dies, Clostridium perfringens multiply rapidly to decay the bacteria. Remember, it's something living dies, it has to be decayed so that the resources can be reused. Everyone can recognize the smell of decaying vegetation. And it's caused by Clostridium perfringens. Everyone can recognize the smell of a dead animal, and that's caused by Clostridium perfringens. If you've ever had the misfortune of being around someone that's been dead for a while, that very unique smell, actually it's close to the smell of a dead rat, is because of the Clostridium perfringens. But basically it's 5 to 9 for that bacterium to grow. Bacterium is singular, bacteria is plural. Time. Bacteria reproduce by going crazy, having kids. Temperature. Most bacteria grow best 41 to 135 degrees, which is why that's so important to keep food out of that temperature range. Bacteria vary in their requirements for oxygen. Billions of years ago, when life began on this planet, we've proven that the methane in the air and the lightning going on produced the first organic substance, which billions of years later led to a small strand of RNA, of nucleic acid, and billions of years later it led to a way for that RNA to enclose itself in an enclosure for protection. Billions of years later it learned how to reproduce, leading to the first life on Earth, which is single-cell animal organisms, and which means bacteria and algae. Bacteria and algae have been there millions of times longer on this earth than we have. They've been there from the beginning. We're just a relatively very late newcomer. So, who knows more about life, bacteria or us? Aerobes, of course, need oxygen. Listeria is an example. Anaerobes only grow when there is no oxygen. It just it was born that way, baby. Botulism is an example. The most potent neurotoxin known to man. Scientists have proven. Botulism can be spread in several different ways. bacterial spores, which cause it. I know somewhere else in this course, I'm going to explain what spores are and how that works. Common in both soil and water. That spores are a way that a lot of organisms reproduce, and when conditions are horrible, can protect themselves from the environment until favorable conditions allow them to become alive again. Well, they're more alive again. Foodborne botulism is when the food, okay, like if a can gets dented, there's no oxygen environment pretty much in that can, but the botulism can produce the toxin, which is why you never buy it except for delivery. Cans which might be dented or something like that. Facultative cells grow with or without. Examples are salmonella. Moisture, water. Life as we know it requires water. Most everything, plant, animals, or water. Their nutrients must be received in a water solution. The availability of water is described as water activity. Water activity goes on a scale from 0.0, meaning no water, to 1.0, meaning pure water. Water activity, AW, I refer to as the factor. Now, foods with a water activity of 0.92 or more contain sufficient moisture for rapid bacterial growth. The AW is not 0.92, meaning a whole lot of water. Ain't nothing happening. Technically, AW is the vapor pressure of a substance, pure water at sea level. What is vapor pressure? It's the pressure above a substance, water in this instance. For example, if you pour water into a pan, it'll have a certain vapor pressure above it. But if you heat up the stove and heat the pan, then there'll be higher vapor pressure because the substance is going from a liquid to a gaseous state, which increases the pressure above it. They take water by moving it across the cell membrane, osmosis. What is osmosis? Now, picture for me, if you will, as Prince says, a U tube, okay? It looks like a U, okay? It's glass, got tube in it. Now in the middle at the bottom, you put a semi-permeable membrane. What is that? All cells have semi-permeable membranes. That means certain substances can move back and forth and some can't. But cells have to have a way to move water, other substances back and forth, and this semi-permeable membrane is the way that it's done. Now, we're looking at a glass tube. Picture this in your mind. And at the bottom of the U, we have a semi-permeable membrane, such as what cells have. Add water. I don't know, so it's halfway on each side, okay? So we got U-tube, semi-permeable membrane, water. Now, on one side, add 1 gram of sugar. On the other side, add 2 grams of sugar. Okay? So we start off with 1 gram of sugar and the same amount of water on one side, and 2 grams of sugar, same amount of water on the other side. Now, obviously, in terms of physics, the side containing the 2 grams of sugar is obviously going to be a lot heavier than the side containing only 1 gram of sugar. So, wouldn't it make sense that since it's heavier, the water level on the side with 2 grams of sugar would lower, and the water level on the side with 1 gram of sugar would rise? Well, guess what? That's not the way it works. It goes the other way. The column with the heavier substance, 2 grams, will rise, and the side with the lower weight, 1 gram of sugar, will lower. It works against gravity. It doesn't give a damn about gravity. I'm going to do it this way. Osmosis. What's happening here, it's equalizing the amount of solute, being what's in the solution, water. The water levels will move until each side is 1.5 grams of sugar. And it doesn't really give a damn about gravity. Yes, it affects it, but the magic of osmosis is that the amount of solute in a solution will cause the action across the membrane, which is, well, cells do it all the time. They know how to do it. Okay. Water movement mechanism. Well, there's a water gradient, meaning pressure gradient, moves from a higher water activity outside of the cell to lower activity, meaning that it's a pressure thing, but then it's controlled through osmosis. When water activity outside the cell, basically pressure, is low enough, osmotic stress, in other words, the cell can no longer use its main trick in the book of osmosis to get stuff in and out of the cell. Since the cell can no longer take in water, it definitely can't grow and reproduce, and with enough, osmotic stress will kill it. The microorganisms are not eliminated just because they're unable to reproduce, unless this goes to the extreme. If an organism copes with osmotic stress in different ways, try the different growth limits for each organism. They all deal with it, adjust to it differently. Some types of most yields have figured out how to live with very low water and acidity level. Each organism has a specific water activity at which it can no longer do its thing, baby, and stop growing. Long as crop developers keep the water activity below this limit, the microbe in question can't replicate to high enough levels to cause infection or illness. Now, on the x-axis going across, we see 3 different pH levels, less than 4.6, between 4.6 and 5.6, and greater than 5.6. And on the y-axis going up and down, you'll see the values, 0.92 or less, 0.92 to 0.95, and greater than 9.5. Now, let's look at values of 0.92 or less and pHs. Well, obviously, if it has an AW value of less than 0.92, bacteria can't be bacteria and it's a non-TCS food. When the water levels between 0.92 and 9.5, depending on the pH, it may or may not be TCS food. If pH less than 5.6, you don't need to worry about it, but over that, then PA, which means product assessment, is required whether it's a TCS food or not. When the water activity value is greater than 0.95 and the pH is over 4.6, it's very well could be a TCS food. So when the pH is 4.6 and over, and the water activity is over 0.95, then you have to pay particular attention to what's going on with that food. Now, bacteria, like all animals, need shelter from the environment. Bacteria commonly use cuts, scrapes, sinus cavities, any place to hide, to live, and it's always warm and moist there, and there's always food there. There are two types of glands which secrete something through the skin to the outside. The first is the normal sweat gland, which is a crine gland on the right. It's basically a tube through the epidermis, and when your body is overheated and needs to cool itself, it will secrete water with little sodium through this tube, or eccrine gland, or basic sweat gland. Now, there's another gland which secretes substances to the surface of skin. It's called the apocrine gland. Apocrine glands secrete substances through hair. In other words, it doesn't just flow out like in the crine gland, like sweat. It goes through hair and is distributed that way. Apocrine glands in the skin, they're only on certain places of your body. Eyelids, ears, armpit, groin, the areola around the breast, that's where they are. When you see actors in romantic scenes in the movie kissing someone's eyelids and ears, that's why. Apocrine is being secreted there, which is a pheromone, and it's an interspecies communication, sexual arousal, hate, fear, different things. And of course, the two main areas of the apocrine: hair on the body, which is also well known for getting kissed, hopefully as much as possible. But apocrine is interspecies communication. All species have an interspecies communication, which is not known. In other words, they didn't say it, they didn't show it, but it was communicated biologically without your knowledge. Fear, hate, love, sexual desires, confusion. We communicate with each other without communicating. You can sit by someone in a movie theater, not look at them at all, but if you're sexually attracted to them, they will subconsciously know it. Don't have to say a word, don't have to look at it. Hey, this point, you can't hide it, except by getting rid of it. Apocrine glands in the skin are liquid producing glands, and guess what? The human body, the pheromones that we humans secrete through the apocrine glands via hair is a very mildly sweet substance. Well then, why do we smell so badly? Body odor, that's not us. We didn't do that. Apocrine glands secrete a substance which certain bacteria love, and when the bacteria go in there, oh yummy, they produce what's known as body odor. So, I don't like to walk around as a harborage for bacteria. There's several reasons. One, it eliminates apocrine, which means no one knows what's up with me. Fear, hate, subconscious sexual arousal, stuff like that. Second, I don't want all this bacteria hitchhiking around making me smell bad. So twice a week, if it isn't hair on top of my head, it gets shaved. Now, guess what? I haven't bought any personal deodorants in over 25 years. Nope. Do I use deodorant? Nope. It's expensive, competing smell. Don't need it. Why? I don't have apocrine glands secreting pheromones that bacteria like to eat. So if you eliminate hair in these areas, may be necessary to, in the morning, run a swallow or some alcohol to get rid of a little bit of bacteria. You no longer require personal deodorants and impressments. Do the scientists that are developing these deodorants know this? Of course they do, but the person paying them $150,000 a year, if they brought that up, they would be looking for another job. But all scientists know this, so don't expect to date a scientist and find deodorant in their bathroom. Apocrine gland, hair follicle, continuously. Well, they're not active until puberty, but then after puberty, that's why little kids don't need deodorant. But after puberty, then they're sexually functional. So then the sexual pheromones for communicating that I desire you become active. When there's some kind of emotional stress, fear, hate, confusion, sexual desire, it creates an emotional stress, and it makes the cell walls contract, and it emits fatty secretion to the skin, where the local bacteria that love it break it down into odorous fatty acid. The pheromone communication still works, but the bacteria that love to eat it causes this horrible odor. So if you shave all the hair on your body, well, it might be once a week, twice, possibly quick swab of alcohol, there would be no interspecies pheromones, which cloaks your emotions much better and eliminates harbors for bacteria to make you stink. Now, bacteria reproduction. For bacteria to reproduce, it's just extremely simple, straightforward, very few problems involved, if any. They just split into two. Okay? Now, that's asexual reproduction, meaning one sex. Earthworms can reproduce sexually or asexually. If another earthworm is not around, it can reproduce by itself. But if two earthworms happen to meet while they're crawling around the earth, they exchange genetic material, impregnating each other with them, and they can reproduce sexually. Sexual reproduction. Boy, now I know everyone's undivided attention. But sexual reproduction is just fraught with huge problems. You could get eaten, shot, sued, all kind of dangers with sexual reproduction, which those who produce asexually, like bacteria, don't have to face. But it's an extremely complicated process, not only to pull it off on the higher level, but for the cells to pull it off. But it leads to greater diversity. That's why sexual reproduction can lead to, I got a kid with red hair, got a kid tall, got a kid short, Two bacteria, no, they're clones. The only way to lead to diversity is through sexual reproduction, so that the genetic material can, through many processes, be intertwined with the other person's to produce varieties. And for one cell to become two goes through an incredible process, and billions of your cells go through this by the second without your knowledge or permission. Interphase, prophase, metaphase, anaphase, teleophase, ta-da! Two. Now, at each phase, cells have what they call cyclin. Cycline. It's a substance that builds up as the cell goes through the various stages of cell division. And if something starts going wrong, the cycline production will be lower. And at two stages during cell reproduction, if it detects something might go wrong, the cell will willingly self-destruct. If it thinks something's going wrong, it will self-destruct. Compare that to the human way of doing it. I don't give a damn if it's horrible for everyone involved. Divide anyway, It's ridiculous. Phases of mitosis. Interphase, chromosomes duplicate. For one cell to become two, it has to duplicate the chromosomes. And prophase, chromosomes condense and become visible. Normally they're all wound up together so tight that it's hard to see under a microscope. But when they're unwound, like a snake uncoiled, then they become more visible. Spindles form, which starts at an outer side of the cell membrane and extends to the nucleus so that it can pull the thing apart. Pro-metaphase. Before metaphase, the nucleus membrane breaks apart. In other words, cells have a cell membrane, not all cells, but we do. Cell membrane, which keeps the nucleus safe, and things go in and out of it, just like the cell membrane on the outside of the whole cell. Things go in and out. Metaphase, the chromosomes are copied. Can you imagine this, unwinding something 6 miles long and then having to duplicate it? That is what happens every time a cell in your body replicates. Has to unwind it and duplicate the entire thing. Anaphase, the chromosomes appear two genetically in identical groups. In other words, they're now two visible things, and they move to the opposite sides of the spindle, which means the things that pull them apart. Teleophase, the spindles attach from the outside of the cell to the nucleus, rips it apart, creating two nucleuses. After that happens, cytokinesis, it splits into two distinct cells. DNA in your body. Some organisms have RNA, some have DNA, depending on the organism. Bacteria can have either. Everyone knows we have DNA, but a whole lot of things going on in our cells is RNA. For example, the cells in our body, they have to continue to produce proteins, which keep them alive and also provides the function of that organ. Your DNA has to continually unwind, separate, and be read. When it's read, it creates what's called mRNA, messenger RNA, that goes outside the nucleus and then the ribosomes read that strand and create the nucleic acids, which will lead to the production of the proteins that's required by the cell and by the organ that it works for. A DNA strand is about 6 miles long. Now, if you took all the DNA in your body and glued it together, it would go from one side of the universe, Milky Way galaxy, okay? Now, we're not just talking, Earth, Jupiter, Mars. We're talking the entire galaxy. It would go from one side of the galaxy to the other and then back again. But it has to really be coiled up. You know how when you twirl a string, it gets, condensed? Actually, our DNA is what's known as histones, which is 4 protein cells, and it gets wrapped around that a little bit, and it gets wound and wound and wound and wound and wound and wound and wound until it's down to about 6 microns, which is 0.0002 inches. Look on your ruler and find 0.00023 inches. Thank you. I hope you enjoyed the lecture. It's extremely important to understand bacteria so that you can know when you understand something, you can better deal with it. And, big thing of your job, not the only thing, but is to prevent bacteria in the food that shouldn't be there. And the avenue doing that is to better understand it. Take a break, and I'll see you in the next video.